data, as we saw on one of the objectives on the previous slide, to support official disease status for a country or a zone or an animal, a domestic animal compartment, and facilitate trade. So animal health surveillance generally works really well if there's careful planning and strategy that's transparent and made aware to all the people and groups involved, all the relevant stakeholders. If there are really clear and agreed case definitions and investigation protocols, both in the field and in the laboratory. And working with the data is obviously a really important part of the process, and having clear standards for the type of data that are collected, how they're collated, how they're brought together, how they're stored, what type of databases are used, what types of analyses and interpretation of those analyses and reporting are all need to be done to high quality if the animal health surveillance program is going to work well. And following those activities, the communication and dissemination of the results of the surveillance are really important, and that can be might need to be done differently for different target audiences, for different groups, whether they might be farmers or the general community or governments or government agencies. The type of communication is going to be different in different settings, and having clearly agreed actions that will be taken based on the results of the surveillance, and that being transparent and clear before the surveillance starts is really important. And as I said earlier, the periodic evaluation and review of surveillance is really valuable and important to keep the process working well, because things change over time from the point of view of what happens with the relevant animal populations, how the disease is working, or the context for the disease in those populations. And of course, we need to have sufficient resources for the surveillance program for it to really work well. And in that context, I think it's useful to recognise that animal health surveillance as a sort of discipline, as a scientific endeavour, is relatively young. The first international conference on animal health surveillance was held in France in 2011. The fourth of these conferences is due to be held later this year in Denmark. We are seeing animal health surveillance increasingly incorporated into legislation in countries or states or provinces of countries, and that One Health surveillance as a concept is really gaining traction in lots of different areas, and recognising the linkages of health of animals, people and the environment, and carrying out surveillance with that perspective in mind is increasingly being attempted. And that evaluation is really crucial, that evaluation process for ongoing improvement. And on this slide here, you can see that on an FAO website that the Ethiopian government carried out an evaluation of its animal health surveillance activities back in 2018, which is really great to see. So to talk a little bit about influenza A viruses that include avian influenza, these are RNA viruses that can have a variety of genotypes, and they're classified according to two of their genes, the hemagglutinin and the neuraminidase, and the classification is used according to the H and N with a number. So you can have H1N1 or H5N1, H5N8 are some of the combinations you might be familiar with. These viruses are highly contagious. They spread easily between individuals in the population. They have a high mutation rate. They're effectively changing their nucleic acid all the time, which presents a challenge for maintaining diagnostic tests, surveillance activities and vaccines to all being fit for purpose. influenza A viruses have an evolutionary origin in wild bird populations, particularly groups of wild birds that form large, dense aggregations, large numbers occurring close together. And the two main groups of wild birds that are form natural reservoirs for avian influenza are the anseriforms. These are the ducks, geese, and swans, and the charadriforms, the shorebirds and gulls. And so influenza A viruses, in addition to those found in those natural reservoirs, are also found on an ongoing basis circulating in poultry and in a few mammal species, namely pigs, humans, dogs, and horses. So avian influenza as a disease is a significant threat to poultry production and trade. And high pathogenicity avian influenza, or HPAI, is the form that is the biggest problem. And a subtype combination H5N1 and then H5 with different N types, denoted here as H5NX, poses an ongoing threat to poultry and has done since 1997, resulting in the death of many millions of poultry. Also, avian influenza poses an ongoing zoonotic risk with various subtypes, in particular H's 5, 7, 9, and 10. And the intercontinental spread of HPAI via wild birds is also an important factor that I'll talk a bit about later. So to talk a little bit about the epidemiology of avian influenza, we have these two main pathotypes, high pathogenicity, or HPAI, and low pathogenicity, or LPAI. So high pathogenicity avian influenza often causes severe disease in poultry and wild birds, not always, but often. It causes a systemic infection. It spreads to many different body organs and systems. It can have very high mortality rates, even up to 100%. It's only been observed within the subtypes H5 and H7 so far. And this H5, HPAI, Asian or Eurasian lineage that I'll talk a bit about later is quite a special case in terms of HPAI. So low pathogenicity avian influenza generally just causes a mild respiratory disease or can be completely asymptomatic in birds. It can be any H type, but it's types 5 and 7 that have a known ability to mutate to high pathogenicity. In poultry, it can cause quite changes in production, like a decrease in rate of egg laying and food intake. It can combine or co-infect with other pathogens to exacerbate other infections, and it circulates in wild birds normally, particularly in the anseriforms and charadriforms. So the pathotype designation really applies only to birds and particularly to chickens in terms of causing high or low pathogenicity. So, for example, a H7N9 subtype combination as a low pathogenicity avian influenza was present in parts of East Asia for quite a few years, where it was not causing significant disease to poultry, but on occasional infection to humans was very dangerous and responsible for quite a few human deaths. So it could be have a high pathogenicity to humans, although designated as a low pathogenicity avian influenza. To move on to the HPAI viruses and how they emerge, how they occur. So the classical epidemiology that's been observed is that to have an HPAI event or emergence, we need to have mutation from low to high pathogenicity taking place after the introduction of an LPAI virus to poultry from wild birds. And in particular, the poultry species being chickens or turkeys, perhaps ostriches are the only species where this mutation has been observed to occur. So not ducks or geese that have a closer evolutionary relationship with the natural hosts. And an exception, however, is this Eurasian lineage of H5 HPAI, and specifically a lineage designated as influenza A, goose Guangdong 196. And this particular lineage has persisted in poultry for decades, and occasionally shown prolonged circulation in wild bird populations. So avian influenza as a disease is increasingly seen as an important disease in poultry, has an impact across society in some countries. Hundreds of millions of poultry have been killed due to infection or cull as part of control measures due to this Eurasian H5 lineage, with costs into tens of billions to the global economy. And we see changes in distribution with uncontrolled spread, endemic infection in at least 6 different countries, and on an ongoing basis, emergence of new clades of this H5 lineage and creating waves of infection. In recent weeks, there's a wave of infection moving through North America, through Canada and the United States. We've seen movement between Asia and Europe and Asia on a number of occasions, periodic introduction to Africa. So it's really a global problem, although this particular lineage hasn't, as far as we know, reached South America or Australia. Avian influenza also has, as I said before, a zoonotic, can be a zoonotic disease, and has implications for pandemic preparedness. Just highlighting the importance of the disease, and it's useful to understand that poultry as a protein and meat source is becoming increasingly important, and that it's estimated that poultry meat will become the largest meat sector in the world in the current decade. So now to move on to how surveillance for avian influenza, how it works, how it's carried out. I'm going to talk first of all about avian influenza surveillance in Europe, and starting off in how the surveillance works in poultry. So a type of surveillance referred to as scanning or passive surveillance is really the front line, or the first type of surveillance that occurs in Europe. And this is surveillance that relies on the detection of clinical signs of disease or other production indicators like reduced feed intake or reduced egg production being a trigger for a disease investigation, to then result in collection of samples that are tested for the presence or absence of avian influenza. So it's an ongoing activity, monitoring the health of birds and resulting in investigation if there's sufficient evidence to suspect avian influenza as a disease. In addition, there's a legislative requirement for all EU member countries in Europe to carry out serological surveillance for evidence of exposure to H5 and H7 viruses, so looking for antibodies. And if those antibodies are found, then the action that's required is follow-up sampling to determine whether the poultry flock might be currently infected. And if infection is found, then there are further actions that are required immediately, involving restrictions of movement of those birds and restrictions on what can happen to them. So to move on to surveillance for avian influenza in wild birds in Europe, all EU member states are required by legislation to carry out surveillance referred to as passive, or the surveillance of birds that are found dead for H5 HPAI. And there are instructions or guidelines for how this can be carried out, set into European or EU legislation, including a list of target species that's based on previous detections of H5N1 HPAI across Europe. Some European countries also carry out surveillance for avian influenza in apparently healthy wild birds, And so this can be through capture, sampling and release, through environmental sampling, or through sampling birds shot by hunters. So the results of the surveillance for avian influenza in Europe are collated and analysed and reported on an annual basis in reports made available on the European Commission's website. A couple of front covers of previous reports shown here. And these reports contain information about the relevant bird populations, so wild birds and poultry, about what sampling has been carried out in those populations and what detections have been made. And there are various ways of displaying that information in tables and graphs and maps. And just a couple of maps here. The map on this slide shows for wild birds found dead or sick, with the number of birds per geographic area that have been sampled, indicated by the colour shading. And then if there's been a detection of an H5 or other LPAI virus, they're shown by the coloured circles on the map. And a similar format of map here, but for poultry. Again, the colour shading is an indication of the intensity in terms of number of farms sampled per unit area, and then any detections of H5 or H7 viruses are shown as the number indicated by the size of the circle shown in red or green. So something of interest is the interface between poultry and wild birds with relation to avian influenza. And in the reporting at the European level, there are various analyses carried out, and I was involved in some work looking at this a number of years ago, where we looked at temporal association between detections in wild birds and poultry. So there's a graph at the top here across a number of years with the number of detections in wild birds per week shown as in blue, and then detections of particularly or specifically of H5 low pathogenicity avian influenza, and then detections in wild birds shown in blue, and detections in poultry shown in red. And we see, if you look at this graph, where there's some activity, some detections in wild birds, quite often they were followed by a detection in poultry. So a peak in blue, quite often seen followed by a peak in red. And if we aggregate those data together on a seasonal basis, so put them all together here, this graph here shows low pathogenicity H5 detections. We see a clear seasonal pattern in wild birds where there's a peak every autumn. So September, October, November, Northern Hemisphere autumn, and this seems to be then closely associated with higher detections in poultry in November and December. Whereas in contrast, if we did the same format of this graph shown over here on the right for low pathogenicity H7 subtype, we didn't observe the same kind of pattern at all. And this is really useful to observe, to help inform the focus of surveillance in future years to improve its efficiency and effectiveness. So just to summarise the European avian influenza surveillance situation, so it's required by legislation. There are actions required to be taken if detections are made in certain types of virus in poultry, such as movement restrictions, and in the case of HPAI, culling of birds on infected premises. And with wild birds, there are movement restrictions that relate to poultry and other live birds in a zone around where the detection's made. The surveillance provides ongoing monitoring of trends and patterns of avian influenza over space and time, and different bird species and virus subtype. And it informs health risks to poultry, to humans, and to wildlife. Increasingly, avian influenza is recognised as an important process as a threatening process for some wild bird populations. and this information contributes to policy development in this area. And a few scientific publications in relation to that are shown here, and I'd be happy to share later. So to move on now to the Australian situation, it's quite different in some ways to Europe, and there are also some similarities. So Australia has never had a detection of Eurasian lineage H5, HPAI. This is a really key difference. And there are, however, wild bird migration patterns that link Australia to other continents where that Eurasian lineage H5, HPAI is present. So we see the Caradary forms, species of wader, about 10 or 12 wader species with total population numbers into several million, move between parts of Eastern Australia and East Asia right up into Northern Russia every year, and pose a potential pathway for movement of HPAI into Australia. However, the Anseriforms, the duck, geese and swan species that occur in Australia, don't show that same type of migratory pattern, although there are some species that have distributions through Australia and into the Indo-Malayan archipelago. So ongoing surveillance and sequencing of viruses found in wild birds has revealed periodic virus exchange between Australia and Eurasia. And in Australia, the HPAI outbreaks in poultry have been small in number. There's been 8 altogether ever recorded. They've all been of the H7 subtype, and those outbreaks individually have all been relatively well controlled and limited to only 1 or 2 or 3 infected farms. So the way surveillance for avian influenza works in Australia is that all avian influenza subtypes are nationally notifiable if they're detected in poultry. There's various other nationally and internationally notifiable poultry diseases that are looked for through ongoing surveillance of the scanning surveillance or passive surveillance types in poultry. And rapid reporting of any detections of those diseases is required in the relevant legislation. And Australia has national reporting systems for any for surveillance in domestic animals and wildlife. So to move on specifically to the surveillance for avian influenza in wild birds, and describe a bit about how it's different in Australia. So a key feature is that the ongoing surveillance in poultry tends to have very few detections of avian influenza in Australian poultry. The poultry population compared to other continents is relatively small and maintained at fairly high biosecurity. So this situation, combined with the level of isolation of avian influenza viruses in Australia compared to other continents, means that if Australia is going to be able to monitor and track the ongoing evolution of avian influenza viruses on the continent, surveillance in wild birds are the most sensible population to look at. And so this is done through targeted surveillance on a risk basis, sampling locations that are close to poultry farming and human populations, involves the sampling of healthy live birds, bird shot by hunters, and environmental sampling, and undertaken by a range of different state and territory governments and other partners. And so this activity is combined with so-called general or passive surveillance, which is the investigation of significant or unexplained morbidity and mortality events in wild birds that can be reported and submitted through members of the public or a range of different veterinary organisations and zoos and universities. And this activity is primarily present to enable or facilitate the detection of HPAI should it be introduced to Australia through the wild bird population movement. So in Australia, a key difference in the reason for doing that active surveillance in wild birds is, as I said earlier, to monitor the ongoing evolution of the avian influenza strains, because they do change over time, and every few years the diagnostic tests that are used have reduced sensitivity and sometimes specificity. So the ongoing surveillance allows maintaining fit-for-purpose diagnostic tests. This is done through sequencing of detections and through laboratory network activities. And it also allows ongoing monitoring of genetic variation and diversity of avian influenza viruses and the maintenance effectively of a sequence library. And I'll talk a bit about that application in a minute. And again, as I said earlier, because of the because detections in poultry are relatively rare, the sampling in wild birds provides throughput for the laboratories in terms of maintaining capability and capacity, throughput of samples. As for in Europe, the sampling provides enables or informs risk assessment and management for poultry, for human health and wildlife health. It also contributes to international obligations around reporting, and there's an important biosecurity awareness and communication component. So just a slide here showing some of the way the results are collated and reported. So looking at different host types. So here's a wild duck species, Pacific black duck, compared to an anseriform, compared to a charadriform here, the redneck stint, and patterns of detection of avian influenza are recorded according to geography and over time. And this helps inform the risk. A couple of slides here just showing a collection method of healthy live birds using cannon netting, so a large net fired over groups of wild birds here. These are wild whistling ducks. Here, a slide showing a species of native goose in Australia, magpie goose, that occurs in Australia and parts of New Guinea, Papua New Guinea and Indonesia. It's actually evolutionary quite distinct from the other geese species that we're more familiar with on other continents. And the application of molecular epidemiology, I mentioned earlier, has particular relevance in Australia. So this is a phylogenetic tree here showing the results of sequencing of the H7 gene. So we see H7 genes from avian influenzas form separate clades from the Americas and Eurasia and Australia. And so maintaining this Australian library through the sampling of wild birds means that if there's a detection in poultry in Australia, the virus can be rapidly sequenced and compared to that background library from Australia and other continents. And this then where the sequence from poultry sits provides really useful evidence for whether the outbreak in poultry is more likely to have occurred from a local spillover, so the introduction of virus from a wild bird in Australia directly into a poultry farm, or the alternative hypothesis would be a human-mediated introduction of avian influenza, perhaps from another continent. And so there's been further work in this area just in the last couple of years, looking at better understanding the global picture in relation to Australia and movement of avian influenza genes of various subtypes, and so effectively the movement of viruses between Australia and other continents. And I'll talk a bit more about this in my last presentation on this course. There's a paper on this work available currently as a preprint and in press with PLOS Pathogens, looking at the relationship between avian influenza viruses in Australia and other continents, and also within different locations in Australia, looking at the patterns of movement of these viruses through the wild bird populations. So just to review the situation, avian influenza surveillance in Australia. So like in Europe, it's required by legislation. There's the ongoing surveillance triggered by disease, clinical disease in poultry, and with action required to be taken if there's the detection of certain types of virus in poultry. So any H5 or H7 will trigger action involving movement restrictions, disease investigation, and potentially culling if it's a high pathogenicity virus, and detection of high pathogenicity virus in wild birds, which triggers actions around movement restrictions for poultry or other live birds in the associated zone. The surveillance activity involves the monitoring of trends and patterns. It informs health risks in poultry, in humans, and increasingly to wild bird populations, wildlife health, and contributes on an ongoing basis to policy development. And again, here are a couple of publications that I'm happy to share after the presentation that give some background to that. That concludes my talk this evening. Thank you very much for your attention, and I'm happy to take some questions.
Thank you for the presentation, Andrew. Are you there?
Yes, thank you. Yep, I am.
Oh, good. Just checking here in the chat to find out if we have any questions about avian influenza. A lot of people thanking you for the presentation.
Yeah, I don't see any questions so far. Hopefully most of the messages were clear.
Yeah, I don't see any questions at the moment. Okay, because if no one has any question, we might move to the next presentation.
I can maybe make just one comment, I suppose, that to sort of link this presentation to my final one on the course that I think is coming in a week's time. Well, I see someone has a question. Alexa? I thought I saw someone, Alexa, saying they have a question. Did you want to?
Oh, yes.
Yes.
How does all this affect endangered species?
Yeah, sure. I mean, so there's quite a bit of variation depending on the situation. I mean, I guess historically, the low pathogenicity avian influenza forms are really circulating all the time in wild bird populations at some equilibrium or balance. And so for any population, they're unlikely to be exerting any significant effect. However, it is different for high pathogenicity avian influenza, and we've certainly seen, especially in recent years, some mass mortality events of thousands of individual birds, and sometimes including birds that are endangered or species that have restricted populations. And this has been most evident just in the last couple of months, actually, has really come to prominence, and I'm happy to share more information with people who are interested. But a couple of examples have been in the Middle East, in an area of Israel that where there's supplementary feeding of a crane species in a wetland area, and avian influenza of high pathogenicity form was presumably brought to that area by other wild bird species, probably a duck species. And it seems that these cranes were really quite susceptible, and quite a significant proportion, I can't remember or don't have the details in front of me, but a significant proportion of that species population in that area has died because of the high pathogenicity avian influenza in that location. In the same time period, so just in the last 3 or 4 months, there's also been a significant die-off of a goose species, barnacle geese, in the United Kingdom near the sort of Scottish and English border. And I think it's 20% of that barnacle goose subspecies is thought to have been killed by that outbreak. And there's been also some significant mortality events in Europe of some wader species. I think red knots or great knots in some parts of continental Europe. So yeah, I think the impact of high pathogenicity avian influenza causing problems for some endangered wild bird species has probably increased in recent years, and certainly has been observed more so in recent years. So I think there's no doubt that that's it's increasingly recognised as a problem, not just for poultry and sometimes humans, but for wild birds as well.
I was just going to say, you have another question. You were just mentioning humans, and someone is asking if the virus can be transferable to humans, and how can we prevent it?
Yeah, so at a basic level, any influenza A virus could potentially infect humans. Most avian influenza viruses are not well adapted to infect mammals of any species, including humans. And it's certainly possible to, when an avian influenza virus causes an outbreak anywhere or is detected in a new place, sequence that virus and then look for particular markers of the genetic code that indicate its ability to infect mammalian cells. So you can kind of do a risk assessment, and it's part of what surveillance can do, is provide you with some insight into that risk of different viruses, the ability to infect people. So there's certainly potential there from any avian influenza virus. There are certain subtypes and certain lineages that seem to be able to infect people more easily than others. And how can it be prevented? I mean, difficult to absolutely prevent chances of human infection. I mean, a few things that certainly can be done in situations where people are responding to outbreaks. So say in the wetlands in Israel, when people are to help reduce the mortality on other wild birds, it's really advisable, it's helpful to go in and remove the carcasses. So the carcasses that have a lot of virus don't stay in the environment. But for people doing that, wearing appropriate PPE, personal protective equipment, so masks and face masks and eye goggles and gloves, etc., just to reduce the chances of infection. And obviously the same for people working in poultry that are infected. So those sort of physical barriers. And then also people can actually can be treated with antivirals as a precautionary measure. So the sort of Tamiflu type drugs can be used as in a preventative way for people who have a higher chance of exposure.
We're having a few more questions. I don't know if you can see them or not, but I'm just going to read them. At what circumstances viruses of wild birds transfer to hens?
Very interesting, yes.
Yeah, so that interface between wild birds and poultry and chickens or hens, the circumstances for that are variable, and different settings, it happens in different ways, and really is quite a big topic. I'm trying to briefly summarize. I guess it depends particularly on the way the hens or chickens are kept. So whether if they are intensively housed, so in a shed with netting and there's physical separation between those birds and wild birds, then infection can still occur, and it's often indirect, such as feces from wild birds that could have viable virus moving into the shed on the boots or equipment of poultry farm workers. In other settings, so poultry that are, say, free range, out wandering free in fields or those in backyards, it can just be close contact. So wild birds, wild ducks or geese can just be landing and feeding from the same food troughs where chickens are receiving their own food. So that sort of closer contact is also possible.
I have here one last question. Do you think influenza virus gives a clue for COVID-19 transmission prevention and control task?
That's an interesting question. I think there's different parts to the question, I suppose, in terms of whether influenza gives a clue for COVID-19 transmission. I guess if we're meaning transmission from animals to people, I think the likely circumstances are quite different in terms of the circumstances for COVID-19 virus, SARS-CoV-2, transmission from animals to people probably has occurred in a market setting, but maybe also in harvesting wildlife. I suppose there's some similarities. It's known that some human infections of avian influenza virus have occurred also in market settings, for sure. Prevention and control, I think that, yeah, there are some similarities there in terms of trying to understand that interface where a pathogen is moving between populations, trying to understand what are the particular circumstances where the virus can be more likely to make a jump, and the methods that we have to understand that. So particularly, I think these days applying molecular epidemiology, sequencing viruses in animals and in people, and really trying to understand where and when transmission occurs, and then looking for ways to manage that, whether it's through better hygienic practices and personal protective equipment, that sort of thing. I think there could be some, but there certainly are differences between those two diseases.
That's perfect. Thank you so much, Andrew. I think that's all the questions we have at the moment. Thank you so much again for the presentation, and we're going to be looking for your next presentation and last presentation next week. Thank you so much.
No problem.
Everyone
Thank you very much.
Thanking you.
Thank you. Thanks very much.
Thank you.
Okay. Bye-bye.
Bye. We're now going to move on to our